Welcome to the Jack Westin MCAT podcast with your hosts, Michael Maleka
and Molly Hudash.
Hello, everyone, and welcome back to another episode of our Jack Westin MCAT podcast with your hosts, Mike and Molly. Mike, I feel like it's been so long, my friend. I'm excited to be here, to be chatting with you about actually a really fun topic.
A really cool one too, right? And again, yeah, it has been a while. I think for like podcast episodes, like it was just like last week. But for us, we actually it's been a while. I was away for a little bit, but now we're back. And again, I am so happy talking about this topic today because it's so interesting. We're going to be really focusing today on Mendelian inheritance and some exceptions to Mendelian inheritance. And when people hear the term Mendelian inheritance, like what does that term even mean, Molly?
Yeah. Well, it's all genetics, right?
Really?
It all is how genes are passed on from parent to offspring, basically. But also more than that, like how do the genes that you have correspond to what you look like or how you behave or how your body functions, etc. So we're going to get into a lot of really fun DNA biology. And I'm excited because I got my little DNA molecule all ready to go.
For anyone
I know.
watching, you saw that, but for anyone not watching, she has a like a DNA molecule on her shelf, which I'm low-key jealous of, but either way.
I'll send you one. We'll match.
Yes.
Yeah, super excited. I think a lot of students are familiar with Mendelian inheritance at some point or another. They've learned things like Punnett squares. You might have heard language like genes and alleles and chromosomes and genotype and phenotype and all these fun things. But for most of us, it's probably been a while. And ultimately, this is a concept that is built on a lot. Okay, will you see 40 Punnett square questions on your test? Maybe not.
No,
but
probably not.
This material underlies so many of the really important biological systems that we see and study, right? It's very critical. It's kind of a foundational topic within molecular biology that really builds into a lot of the stuff that we see on the MCAT. So I'm excited. And if, let's say, anyone listening, it's been a while since you've seen the material, let's recap some just basic vocab, okay? Right. Because there's a lot of terminology in this section in particular that gets really complicated, right? What might we want to familiarize ourselves with?
I think let's start with like the basic term that we probably all heard of, a gene, right? Like what is a gene? So a gene is simply just a segment of DNA that carries a trait, right? Traits can be very widely, like they're normally things that we say are physical. So like traits like hair color, eye color, amount of facial hair. These are things that we call traits. So a gene is a segment of DNA that carries a trait. The gene for hair color, the gene for XYZ. And that's when we see the gene, it's this whole segment. We again, what's normally confused with, like what like is an allele, right? Because a lot of the times these are used interchangeably, but an allele is a certain version of a gene. Because we know, like a gene is a segment of DNA that's a trait. So I could have the gene for hair color, but we know, just in observing everyday life, people have many different hair colors. So that means they have different versions of the gene. The version of the gene that's black hair, the version of the gene that's brown hair. Again, for context, I'm very much simplifying genetics right now. But again, it's not just one gene.
The point.
But it carries the point where it's So thank you. But it's like, it's the gene for red color, the gene, and these are different alleles, different versions of the gene. So it's the gene for hair color, and then you have the allele for brown hair, the allele for black hair, and because we're humans, we have two copies of the gene or two alleles for every gene. So these terms, again, I think are a lot of times used a little bit interchangeably when they're not quite saying the same thing. So I think it's important to know what these two terms are.
Yeah, absolutely. Right. So we've got genes, we've got alleles. I think another word that gets thrown in there that gets kind of confusing is a chromosome, right? But if we think a gene, and I always said a gene is general, right? Hair color is general, right? You could have blonde hair or brown hair or black hair or red hair or whatever, right? Like gene is general. A chromosome is going to be a collection of genes that are going to fall within the same actual molecule of DNA, right? So ultimately, right, we talk about chromosomes. Humans have 46 chromosomes. We have 23 pairs of chromosomes. You actually have two chromosomes that have the same general traits that are within them. Keep in mind, we talk about traits, we talk about genes. These are just literal sequences of nucleotides, right?
Hmm.
We're talking about the information they encode. But when we talk about a chromosome, these are long groupings of genes that fall within the same continuous molecule, right?
Hmm.
We have 23 unique sets of genes that are on separate molecules, but we actually have two copies of each chromosome. One comes
Hmm.
from mom, one comes from dad. We call them homologous chromosomes.
Hmm.
If we talk a little bit about cell cycle things like that, we might get into that as well. But you have two copies. This brings up really what we're going to talk about today. If you have two copies of a particular gene and therefore two alleles, two variants of those genes, what if they're different? If
Mm.
they're the same, right? Like brown eye allele and brown eye allele, like, okay, you're probably gonna have brown eyes,
Sure.
right? Blue eye and blue eye, cool, you've got blue eyes. But what if you have one of each, right? That
Ooh.
is really something that Mendelian inheritance is gonna focus on, right? And
Hmm.
so we need to make our other most basic distinction, right? Which is the difference between a genotype,
Hmm.
which is the type of genes you have, right? Or
Right.
really more specifically, the alleles that you have for that gene. Again,
Hmm.
one comes from mom, one comes from dad. Just literally what alleles do you have?
Hmm.
What combination? Versus the phenotype. And that is
Hmm.
the like observable outcome of those two alleles, right?
Hmm.
So typically it's going to be a physical characteristic. Again, we're being pretty simplified when we talk about hair color, eye color, but it's anything. Some
Hmm.
genes even encode more so like patterns of behavior and mood. It's
Hmm.
something that's observable, okay?
Right.
And so genotypes is very specifics to the genetics, right? Phenotypes is how basically what is the consequence of those
Right.
alleles that you have.
Right, right. And so it's like kind of like distinction that you were mentioning before. The genotype might be one allele for brown hair, or brown eyes, one allele for blue eyes. So that's the
Mhm.
genotype, brown and blue. But then what's the phenotype? What do you actually see? Right? That's the question. And that's kind of really brings us into kind of the foundation, really the father of genetics, which is Mendel. Because that was kind of the question that Mendel was like trying to figure out. Gregor Mendel. I believe he was like a monk, like
He
in
was a monk.
like he was like a monk, which I thought was so sick. He was like a monk, like,
Interesting.
and he was like just, I mean, he was like trying to figure out, like, he had these like pea plants, and he wanted to see, like, hey, if I cross these peas in a different way, what do their offspring kind of look like, right? They might have a certain genotypes, but what is the phenotype that we'll see? And it was from Gregor Mendel, who's kind of the father of modern genetics, where we kind of derive three laws for inheritance, right? And these three laws, the first one is the law of segregation. Right? This law of segregation says about how each parent passes down one allele or one version of the chromosome. Because, like you said before, we have 46 chromosomes, but there are 23 pairs. So there's like the chromosomes are labeled like 1 to 22 plus the sex chromosomes. So like one technically 1 to 23. And you get 23 from mom, 23 from dad. And so we only get one copy of each. That is the law of segregation. It's
Mhm.
important when we talk about Gregor Mendel's laws that we also note that some of these are a little bit overly simplified because we know in reality things get a little bit complicated. Things sometimes don't happen the way that we think that they're going to happen. And so while normally, yes, we only get one copy of an allele from mother, one copy from father, things happen where that might not be the case, where sometimes because of a process known as non-disjunction that happens during meiosis. Again, we're not going to dive into cell cycles too much today, but something happens where instead of one copy of the allele going into those gametes that will be fused to form the human, um, two are taken instead, two copies from allele because of a process of non-disjunction where they don't actually separate. The two sister chromatids don't separate properly, and then they actually go to the same gamete. So in things like that, that's when the law of segregation is a little bit violated, where instead of one copy, you get two. And some impacts of this are things that like trisomies or monosomies. Like a very common trisomy is trisomy 21, which is Down syndrome. So like this is like these are the kinds of things where that's when the law of segregation wasn't exactly like followed, I would say. But again, for the most part, we follow Gregor's laws, where the law of segregation, for the most part, we get one from each parent.
Yes.
Uh-huh.
Yeah. So I mean, keep in mind, if you want to create a new human with 46 and you have two parents that are each going to contribute, you need them both to cut their genetic information in half so
Hmm.
that they can combine to form another human with 46, right?
Right.
This is very much getting into meiosis, right?
Hmm.
In the process by which you take one cell and make four offspring cells, right?
Hmm.
It's basically sexual reproduction of a single cell. It happens in our germ cells. And yeah, you're right. There's lots of opportunities for things to go wrong. Non-disjunction can actually happen in meiosis one or two.
Or two.
So there's a lot of different options for how you could mess with that process.
Hmm.
But yeah, they cause chromosomal disorders, right? You've got too many or too few of a particular chromosome. Law of segregation, though, we're assuming ideal situation, every gamete gets one copy of
Hmm.
each gene from a given parent, right? And the zygote or the offspring will get one copy from each. We're happy we're back at 46 chromosomes,
Right.
right? Also in the realm of meiosis, right? Mendel's second law is the law of independent assortment. This we
Hmm.
actually talk a lot about in
Hmm.
meiosis. It's the idea that if you are looking at, let's say, a given parent, right? During the process of making a gamete, your parent your parents got chromosomes that came from their mom and their dad, right? You might have, according to the law of independent assortment, when all of these chromosomes line up in the center of the cell and get ready to split, you could have, in this case, your grandma's chromosome across from your grandpa's, or for chromosome one, on the other chromosome, you could have your grandpa's and then your grandma's, right? Like every gamete that is
Hmm.
made is going to have a pretty random assortment of chromosomes, some of which came from that person's mom, some of which came from that person's dad. And it's just as likely for you to have a gamete with all of your mom's chromosomes in it by chance, um, as the opposite, right? Basically, it's completely random
Hmm.
how you will line up at that central plate.
Right.
Um, and that's something that's very typical. Basically, what we're saying is if you are located on different chromosomes, I'm
Hmm.
saying you as in genes,
Hmm.
if genes are located on different chromosomes, they're mixed up very frequently.
Right.
That, though, also introduces a possible exception. And again, we
Hmm.
will talk a lot about the exceptions to these laws, where sometimes if you are on the same chromosome,
Hmm.
you don't mix up very often. Okay, we call
Hmm.
that linkage because they are
Right.
physically linked and tethered together. Um, that would be an exception to this law, right?
Right.
And with linkage, we're going to see lots of weird stuff with frequencies of recombination, all that good stuff. It's actually a really fun topic.
It's a really cool topic that like I think it's really cool that they're like how often are they recombined? Yeah, because this is the idea like an example of like independent assortment. It's like yes, like maybe the allele for black hair and the allele for blue eyes, technically like based on law of independent assortment, it should be completely random which one goes where. However, if these two genes are linked, they're very close, maybe, oh, it's if you have black hair, this person also a lot of times has blue eyes because those genes were linked. Those alleles were close together. And so this happens a lot when genes are linked, when they're very close together in the same chromosome. When they're very far apart on the same chromosome, there's this thing of crossing over where a lot of the times because of crossing over, they're not because they're so far away, they don't aren't normally linked together. But it's when these genes on the same chromosome are very close together, that's when they're like, oh, they normally like go as a pair, they go together, then we say they're linked. So they don't really assort independently anymore, which is the law of independent assortment. They kind of like go together. So again, those are the first two laws. Law of segregation, law of independent assortment, and then there's kind of one of the big laws. I think the MCAT of these three laws really focus on this law a lot, which is the law of dominance, right? Where, like we said before, you have two alleles for every gene, one from mom, one from dad. But if you have two alleles, right, one of them has to show, right, based on the law of dominance. One of them will be the dominant allele, one of them will be the recessive allele. Right, so it's the dominant trait. The law of dominance is the dominant trait controls the phenotype. So in our example before, we had the brown eye color and the blue eye color. Well, brown eye color, I think, and this is just generally true, again, very simplified, is generally dominant to blue eye color. So that's why when you have both of them, the person, even though they have an allele for blue eyes, will show a brown phenotype because the brown eye allele was dominant. So this is the law of dominance, that the dominant trait, the dominant allele, is the one that controls the phenotype.
Yeah.
And then there's all these different combinations that we can talk about later, and a lot of exceptions to this rule that we'll talk about. Not right now, in a little bit. It's kind of its own section today, because again, it's really tested a lot on the MCAT.
Yeah, one of
Yeah.
the themes of this podcast is going to be you need to know a lot about Mendel because he was so he produced work that was so incredibly impactful on the
Right.
field of genetics, but also pretty much everything going on with humans is more complicated than this.
That's
Almost
the theme.
everything.
It's
Like
almost
even eye color. My dad has blue eyes, my mom has green eyes, I have brown eyes. Like things are not perfect. It's very rare that
Mhm.
you have a one allele system, but we will give examples of one allele systems. There are a
Which
couple
are
examples that we'll talk about.
they're pretty cool.
But I mean, in terms of how you will see specifically Mendel's laws tested, you could see Punnett squares, right?
Hmm.
Which it's very visual, right? If you have oh, and you know what? We should say heterozygous and homozygous too. If you
Right.
have two copies of the same allele,
Hmm.
what we call it is homozygous.
Hmm.
Homo is same, right?
Hmm.
And zygous for the zygote. It means the zygote has two copies of the same allele. Heterozygous, hetero means different, right? Heterozygous means that zygote or that, you know, basically the cell that becomes a human later on, has two different copies of that allele. So if you, let's say, had someone who is homozygous for the dominant allele and someone who is homozygous for the recessive allele, what would their offspring look like? That's what you'd use a Punnett square for.
Right.
And I think it's easier to see a visual. If you have not seen a Punnett square before, I would Google it so you could see it.
Right. Or maybe, hey,
Maybe
editors, if
we
you
could
want to
have it
show up
appear.
a pun and square, what a pun and square might look like here. I don't know how that would do that visually, but yeah, like it's always heard over like a podcast format to like, hey, this is what it looks like. And you probably anyone listening has probably seen a pun and square before, but if you haven't, look up pun and squares. And again, knowing how to actually form pun and square, because I actually notice this a lot. Like actually setting up a pun and square, sometimes we there's some common mistakes that happen, right? Where it's like not, say someone has like capital B, like how you separate that in the Punnett square. When it's just two, pretty easy. But as soon as you start adding multiple traits, multiple genes, then things start getting a little bit more complicated. So I would say is again, you're listening here, look up a Punnett square, but also how to do a Punnett square with more than one gene is what I would recommend. Like actually figure out a process for that. Again, it's hard over podcasts to actually go through some steps, but I would recommend doing something like that because it's a very common mistake on how to actually do a Punnett square with multiple genes.
Yeah.
Yeah.
Okay, so let's talk some more about dominance, right? We
Hmm.
talked about how oftentimes you have a dominant allele and a recessive allele. If you
Hmm.
have one copy of both, the dominant allele is going to win, right? The
Right.
recessive allele will only show itself in the phenotype if there are two copies of that allele. Okay,
Right.
and that is pretty much what Mendel has accepted. Okay, and
Right.
he found it in a lot of different like phenotypes of a pea plant in shape and color and size and flowers and whatever. In terms of humans, right, when we think of how this applies, we actually see that there's a lot of options, right?
Hmm.
It's not so simple like we've been
Hmm.
telling you.
Hmm.
There are lots of different ways that we can analyze, especially diseases and different phenotypes that create something observable, whether it's good or bad. Okay,
Hmm.
so if we had a particular phenotype that we wanted to track, right, we could use essentially a pedigree. We could look
Hmm.
through generations and learn about how particular phenotypes are passed on, right? Was that allele dominant? Was it recessive? Is it linked to an X chromosome or a Y chromosome? Is it linked to the mitochondria, right? Because it's got its own genome. How? Where is this allele, right? And
Hmm.
the allele of interest is that dominant, is that recessive. There's a lot that we could look here.
Hmm.
And I said the word pedigree because oftentimes when you are in a genetics class, you'll see a lot of pedigrees where they've got squares for the men, circles for the women. You've got different generations shown
Right.
visually in like a family tree. You've got
Hmm.
shaded boxes and unshaded boxes. Just so you know, the AMC has in the past put pedigrees on the test, but just for your own knowledge, the AMC has released a statement that they will not be testing on pedigrees moving forward. There is one pedigree question or pedigree figure
Chart.
in a
Yeah.
passage on one of their exams. But just so you know, I don't want you to panic. If you are not studying pedigrees right now, you don't need to. When you get to
Hmm.
that question of the exam, do your best with it. You'd
Hmm.
probably be able to do more than you think you would.
Hmm.
But on your real exam, you will not see pedigrees.
No. Yeah, no, and I think that's very important that like again, the ideas of pedigrees, like the theory behind it is good, but you will not yet be tested explicitly on pedigree charts. But I think it's cool to talk about, what pedigree charts showed, right? Things like, oh, when we're looking at like a disease or a phenotype, again, it's used a lot for diseases, but again, you could do it on like hair color, eye color, things like that.
Anything, yeah, anything you're
Anything
curious
really,
about.
anything that you can inherit. We say there is a certain dominance or recessive. We can say based on that chart, is this allele dominant? Is this allele recessive? But also where in your genome it is. The term genome refers to the whole collection of chromosomes because there's kind of two places you can find an allele and that we kind of
Mhm.
separate. The first 22 chromosomes, we said there's 23 pairs. The first 22 pairs are our autosomes, or our autosomal chromosomes, and those are the chromosomes that carry most of the traits in your body. The first 22 autosomal. The 23rd pair is the special pair, and those are the sex chromosomes. This is where that XY, XX, or some other combination that's kind of like our client, like Klinefelters, all those things, that's where the 23 chromosome. So when we talk about like diseases or we talk about certain traits, we say, are they autosomally linked? Are they on the first 22 chromosomes or are they on the 23rd chromosome? Because of how unique the 23rd chromosome is on determining sex, it's important that we kind of distinguish them because how it's passed on is very interesting as well. And so we say a certain trait or disease is either autosomal dominant. That means it's a dominant allele on one of the autosomes, or it's autosomal recessive. It's a recessive allele. Like if it's paired with a dominant, it's not going to show up on one of the autosomes. So autosomal dominant, autosomal recessive on the first 22 chromosomes. Sex
Mhm.
chromosomes, we separate a little bit more. So I'll do the first one because I think it's the easiest one. I think it's of the sex chromosome, I think it's the easiest one. Then I'll let you take the other two. So the first is a Y-linked chromosome. Now it's very interesting. There's kind of like a Y-linked, sorry, a trait. And it's very interesting. You kind of have like Y-linked, and it's so interesting to have like Y-linked dominant, Y-linked recessive. You only have one Y chromosome for a male, so you don't really have dominant recessive so often. But a Y-linked chromosome is one that is located on the Y chromosome, and how you know if something is a Y-linked trait is if it's only ever on males. If this is a trait that only men have, like zero women have it, and only men have it, it is Y-linked. Because guess what? Men are the only ones that have the Y chromosome. Biological females do not have a Y chromosome, right? Males are XY, females XX. So a really great hint is if something's Y-linked, if only, and I know it's my extreme language, only males have it.
Yes.
Yeah.
Yeah, because ultimately, when we talk about dominance relationships, we're looking at both alleles and asking which is going to show through. Again,
Hmm.
if we were talking about autosomal,
Yeah.
right, there's no, biological males and females will have two copies of every gene,
Right.
right? That's almost always true, except with the XY chromosome. So if it's
Hmm.
autosomal dominant, right, if you have, let's say, two copies of the autosomal dominant allele, no question, no matter what gamete you give off, it's going to
Hmm.
have that autosomal or that dominant trait. And therefore, if I have two copies of that dominant allele, every single child that I have is going to have that trait.
Hmm.
Because if they have one copy, if they are big A, little a, right, which by the way, a capital letter is to denote dominance. If you have one copy of the dominant trait, you have it, right? The only
Hmm.
time that you would have an autosomal recessive disease, let's say, is if you had two copies of the recessive allele. So that's super
Hmm.
important to understand. Again, with Y, you either have one Y chromosome or you have zero Y chromosomes. So I
Hmm.
love that you pointed that out. There's not really recessive or dominant. It's like if you have that,
You
that's
got it.
it.
You've got
I
it.
also
Yeah.
I feel like it's relevant to say every chromosome, right, 1 to 22 in terms of the pairs, the chromosomes that you get from mom and dad have really the same length. They're the same
Yeah.
size, right?
Yeah.
X and Y chromosomes are not.
Yeah.
The X chromosome is pretty large.
Yeah.
The Y chromosome is tiny,
Tiny,
very,
tiny.
very tiny. And really, of all the genes that are on there, there's really one that matters, the SRY gene, which is literally the don't make me a girl gene.
Yeah,
Like
yeah,
you really
it's
have
so
one functional gene on that Y chromosome, which says I am a biological male, and it
yeah.
controls male secondary sex characteristics, development, etc. The X chromosome, again, females, biological females will have two copies, biological males will have one. The X chromosome now gets really interesting. Okay, so we
Mm,
can have recessive and dominant,
mhm,
right? For a female, you can have one copy of each. Let's say if you had a dominant allele and a recessive allele, of course, you're going to express the dominant trait, but
mhm,
there is a relationship because you have two of those alleles.
mhm.
For a male, though, they
Yeah.
have an X and a Y. We call them hemizygous
Hmm.
because hemi is half. They only have one copy of the X chromosome. So in a female, let's say that we had an X-linked recessive disorder, right? So if you only had the recessive copy of that X chromosome, you would have the disease.
Right.
A female could have a dominant copy and a recessive. We call that a
Hmm.
carrier because
Hmm.
if you have a dominant copy that's not disease phenotype,
Hmm.
your biological female is going to be fine.
Hmm.
That being said, let's say hypothetically, this is me. I have a dominant and a recessive. The recessive is disease encoding. If I had a son, right, I know that my partner would have to donate that Y chromosome,
Yeah.
right? Because it's a son. So
Hmm.
that child has to get one of my X's. There's
Hmm.
a 50-50 shot that that boy gets my diseased phenotype,
Right.
right? Or my diseased allele. And in
Right.
that case, there's no balance. There's no opportunity
Hmm.
for that boy to get a second X chromosome that could balance it out. So
Hmm.
whatever X chromosome a boy has, right,
Hmm.
or a biological male, I keep saying boy,
Hmm.
that is going to be the phenotype that shows.
Right.
Okay, so
Right.
it's
Yeah.
very interesting when we
It's
talk
super
about
cool.
X-linked diseases.
And I think a great example of this, of an X-linked recessive trait that many men have, that not many females have, is male pattern baldness. Male pattern baldness is an X-linked recessive trait. There's a good reason why not many female, of course, not it's not an extreme case. There are many females that do have baldness. Male pattern baldness, though, is so much more common because it's X-linked recessive. When we get
Hmm,
it from a run, so that's why if you want to know if you have a chance to get it, you look at your mother's father, like your grandfather, and so
interesting.
you look, are they bald? Because that means they pass their X to their daughter, right? If they pass the Y, they wouldn't be a daughter. And then your mom has a 50-50 chance to give it to you. And so my grandfather is completely bald on my mom's side. So I'm like, oh no. But yeah, but that's a really great example of why like many men, male parent baldness, because it's X-linked recessive, there's nothing to balance out, whereas females, not as common because they have that chance to cancel it out.
That's so interesting. Color blindness is another really common
It's another
example.
common one too, yeah.
It's another X-linked recessive. And again, it's way more common in males than females for that reason,
Hmm.
because it's not the most common allele.
Hmm.
And yet, if you have that copy and you are a male, because
Hmm.
you only have one X, you've got it,
You got it,
right? It's
yeah.
pretty likely that if a biological female had a copy of the diseased phenotype or the diseased allele, right? So the recessive allele, that they would balance it out with something that's
Right,
dominant. So
right.
very interesting. You'll
Hmm.
notice that a lot of X-linked diseases are more expressive or more expressed in males because of that hemizygous nature. But of course, you can have X-linked diseases impacting females as well. Again, it's just because you have two copies.
Hmm.
Although we'll actually see only one X chromosome is expressed at a time
It's an
in
interesting
female
one,
cells.
yeah.
It's
Yeah.
very interesting. We'll talk about it a little bit later.
Yeah.
But it's interesting, right? We want balance. Males will only ever have one X and that's it,
Hmm.
right? Females at early points in development will actually silence one of their X chromosomes so that
Hmm.
each cell will only express one at a time.
Hmm.
That being said, you can have different X chromosomes expressed in different parts of your body
It's like
depending
totally
on when
random.
that decision was making or made during development. It's super cool. We'll talk about it later, but
Hmm.
X-linked recessive, X-linked dominance, we really just need to be careful because these are sex linked. They are going to show up differently in males versus females based on, do I have a Y chromosome?
Hmm.
If so, maybe I could get a Y linked, but also if we are talking about X linked, how many X chromosomes do I have?
Hmm.
Right? That's something that we need to care about.
Cool.
Also, in the realm of like sex-based inheritance, we could talk about mitochondrial inheritance. This is
Hmm.
pretty rare,
Yeah.
but just so you guys know, when you have fertilization take place and you create a zygote, right, which is
Hmm.
the cell that will become a human, a baby, and
Hmm.
then an adult, etc.
Hmm.
Pretty much everything comes from mom, including the mitochondria. Now,
Hmm.
mitochondria carry their own DNA, and there
Hmm.
are times where mitochondria can carry a mutation in their DNA, right? That could create a different phenotype. Those will always be passed from mom to children, never from father to children, because the dad doesn't, or the sperm doesn't donate mitochondria to the
Mhm.
zygote.
It doesn't really
So it
donate anything except DNA, really. It's
literally, and the
everything,
centriole,
yeah,
and
and
the
the central,
centriole.
but like mostly everything cytoplasmic. Most of the cytoplasm comes from
Yeah.
like all the
And
ribosomes, all those things, like all the
RNA,
ER, like all that stuff
organelles. Yeah.
comes from mom. Dad gives DNA and like centrioles.
Yeah.
That's it. So
That's
anything
it.
mitochondrial, yeah, it doesn't matter if it's a daughter, it's a son, if it's mitochondrial related. Like I have my mom's mitochondria, Molly, you've got your mom's mitochondria. Like it's
Mhm.
our mom's mitochondria.
Yeah. Always.
Hmm.
Okay, so these are the basic concepts within Mendel. Okay.
Hmm.
And even then Mendel was really focused on the autosomal stuff, right?
Right,
But
right.
these are all things that are for the most part in alignment with these laws of segregation, laws of dominance. That being said, and we've been hinting at this all episode,
Mhm.
there are a lot of exceptions, particularly to the law of dominance.
That the
Okay,
MCAT hits on. Yeah, yeah.
yeah, law of segregation, right, and the law of independent assortment, really the only times that those will have exceptions are the things that we talked about, like
Hmm.
things like non-disjunction, when you
Hmm.
accidentally
Hmm.
give one gamete two copies of a chromosome and then
Hmm.
one gamete gets zero.
Yeah.
That's obviously a problem. That's a separate issue. Same thing with linkage, kind of a separate thing. When we talk about exceptions to the law of dominance, we're basically saying that we don't have something so simple as one dominant allele and one recessive allele.
Mhm.
Sometimes we have more than two alleles that control a given phenotype.
Mhm.
What happens then, right? What does the dominance relationship look like there, right? Sometimes we have multiple genes that control a phenotype, right? There are a lot of ways that this gets more complicated.
Mhm.
But before we get into them, one thing I want to make super clear is the allele distribution when we talk about meiosis, all that is the same, right? we're
Mhm.
not we're not saying that these exceptions are functionally different in the way that alleles are segmented into gametes, right? What is different is how the genes or the alleles that you have for that gene control a phenotype. So
Right.
the relationship between genotype and phenotype is different in these exceptions, and that is what we're getting into. It's not
Right.
simple dominance,
Yeah,
basically.
right. Yeah. Law of dominance was basic Gregor Mendel, but now we get to these like, okay, when you have two copies, let's focus on two, like multiple copies of like alleles is like, okay, that's it's so complicated. But the MCAT really focuses on, okay, say you've got two copies of an allele. All right, but now which one takes over? So there's some different types of patterns. So the first one to talk about is incomplete dominance. So let's switch gears and talk about like red and white on like a flower. I think that's a really good like red and white petals of a flower. I think that's a nice and easy one. So in law of dominance, in normal classical law of dominance, let's say red is dominant over white. Okay, so if I have one red allele, one white allele, then the color should be red, right? If the red is dominant. But incomplete dominance, this is the one where, okay, because it's incomplete, there's not one that dominates the other. There is no real dominance. So instead of one overtaking the other, they blend together in incomplete dominance. So if I have red and white in incomplete dominance, I have pink, right? So they blend together in incomplete dominance. So this is not what Gregor Mendel said. This is like a whole exception to the law of dominance. So that's incomplete dominance. But what I notice a lot of times it gets really confused with co-dominance, right? I see a lot of people like confuse the two between incomplete co-dominance. I always think, if we talked about this before, I like thinking about it like cow dominance. Like cow dominance is like, say I've got red and white. In co-dominance or cow dominance, you would have both at the same time, kind of like a cow has black and white spots. You would have some areas that are black, some areas that are white. In our example, you'd have some areas of the flower that are red, some areas in the flower that are white. There would be no blending, but rather it's you see both in a different distribution. So incomplete blending, cow dominance, you've got like the spots, like a cow has like those spots. So that's how I like kind of separating the two.
Yeah. And
Yeah.
it's, you will see both at the same time,
Right.
right? It's not a mixture, which is ultimately pink is a third phenotype. You could be
Hmm.
red, white, or pink. Pink is not
Hmm.
red or white. In this, you are red, white, or red and white, right?
Hmm.
When we talk about, one of the most common applications of this is with blood types,
Sure.
which, by the way, blood types are absolutely
Super cool.
fascinating.
Super cool.
Really, really cool. We have blood type A, blood type B, and there is blood type AB.
Hmm.
Okay. And each one of those alleles, right? Allele for A and allele for B, we'll call them, they tell that red blood cell to put a particular protein on its surface.
Hmm.
Okay, so if you have both, these are co-dominant to each other. That red blood cell just has both. Okay,
Hmm.
there is though a third allele in that system, and this is what makes it kind of fun.
Oh
There's an
no, you
allele
get
that codes for nothing.
yeah.
What we say is the allele that codes for A and the allele that codes for B, those are co-dominant to each other, right?
Hmm.
Neither one is going to be dominant. If you have both, you have both, period. But both the allele that codes for A and the allele that codes for B are dominant to the little I allele, which codes for nothing, right?
Hmm.
When we talk about blood type O, right,
Hmm.
which is not AB or AB, right? Blood type O means you have two copies of the little I allele. You
Hmm.
don't have A or B antigens or proteins on the surface of your red blood cell. You have nothing. It's almost like a
Hmm.
little naked red blood cell, right?
Hmm.
That's why blood type O is the universal donor,
Hmm.
right? Because there's no proteins that your immune system will see on that red blood cell and then attack it.
Hmm.
It's really cool. There's so much that you can get into, but it's a really common way to test codominance. Okay,
Right.
so again, red and white, creating something that's red with white spots, or cow dominance helps a lot. But know that if you see this tested, a very common way to do so is with blood typing.
Hmm.
Um,
Hmm.
and so it's cool. Definitely check it out. Do some Punnett squares with it. Again, you could still use Punnett squares. That
Hmm.
tells you the genotypes that are possible for offspring, but you need to know what's going on in order to figure out how that will control the phenotype. Again, that's
Exactly.
what these exceptions are for.
Right.
Um,
Right.
so incomplete dominance and codominance. We also, these are both different ways that alleles relate to each other. There are actually times where we see multiple genes
Hmm.
control a single phenotype.
Hmm.
Um, and in this case, especially when we talk about epistasis, we talk
Hmm.
about how one gene can essentially mask what another gene is doing. Okay. And the most common or the easiest to point to is baldness. You mentioned
Right.
it earlier, right?
Mm.
Like in our very simplified system, right? You have hair color. Let's say blonde hair, brown hair, right? Like let's just say that those were the two options, or
Hmm.
brown hair, black hair, who cares?
Sure.
Whatever pair of alleles hypothetically you have for that should determine what color hair you have,
Sure.
right? That's an allele for hair color. But what if you have a separate gene that will make you bald, right? The pattern of alleles that you have for that means that you don't have any hair, right? Or maybe even instead of baldness, alopecia,
Hmm.
right? Where you become essentially allergic to your hair starts to fall out in clumps, right? Like it, now you have alleles for hair color, but those are not being expressed because of the action of another allele.
Hmm.
Or another gene locus.
Hmm.
This is also really what's going on with labs. Um, that's how we have white, yellow, or black, chocolate, and white labs or yellow
Hmm.
labs. You have a gene for coat color, and then you have a separate gene for the deposition of pigment. So like you
Mm.
have a gene that codes for what pigment is made to deposit into a dog's hair. But then
Sure.
you have a separate gene that determines whether or not you will put that pigment anywhere that it will stick anyways.
Mm.
So it's like if you don't if you don't have nails, you can't hang a picture, right? Like
Right.
it doesn't matter what
Hmm.
it would look like. If that pigment never gets deposited, you get a white lab, right? Or
Right.
a yellow lab because you don't get that black or brown pigment setting in. So
Even though it
again,
might have the gene for that black or brown one.
yeah. So
Mhm.
you only see the phenotype from one gene if conditions are right in another gene. That's
Right.
really what epistasis
Right.
is. There's lots of different side tangents you can get off there, but that's the most core idea, and it's pretty fun to talk about.
It's a cool one. I think it also like things like those. Like I think epistasis, like as a concept for the MCAT, is tested, but like not I think nearly as much as like incomplete or codominance. But
Yeah.
as an idea, I think if you
Mhm.
understand epistasis, you kind of really are able to conceptualize how interrelated the body is, like how so much goes into really everything, into like skin color. It's not just one gene. There's like all these interacting things, hair color, eye color. There's all these interacting genes. So I think epistasis just as a concept is really great to understand just for the, I mean, I just think for the love of the game, like just for as just a really enjoyable thing. Because realize, oh, it's not so simple as just one gene. Things interact in the body. Another really interesting kind of not exception, but like kind of variation to dominance is expressivity. Right, because like we said, I'm gonna go back to my like simple red and white example of like flowers, right? You would think, okay, red is dominant to white. So if I have a heterozygous genotype, red allele, white allele, I should be red. But variable expressivity refers to the idea that you can have the genotype, but the phenotype can vary in intensity. So
Mhm.
say I've got a heterozygous red, white, I know my phenotype will be red, but variable expressivity will mean that, oh, I have a different shade of red. I could have the exact same genotype, but maybe I'm a light red versus a dark red. So I can have a whole group, say all my offspring, have that genotype. Some will be light red, some will be dark red because of variable expressivity. How intensely does that phenotype show?
Yeah,
Mhm.
it's it's fascinating. Again,
Mhm.
we present a very simple system with
Right,
Mendel, right?
right.
And then we just kind of blow that
Blow it
simpleness.
up. It's like, hey, life a little bit
Right?
more
Like,
complicated.
even if you have true dominance relationships, you might not show the same degree of the dominant
Yeah.
phenotype. And even this kind of goes hand in hand, but from a different perspective, the idea of penetrance,
Hmm.
right?
Right.
Penetrance is essentially, right, rather than like to what intensity do you express the let's say dominant trait, penetrance is, does it matter that I have the dominant trait, yes or no? So there are times where we have dominant alleles,
Hmm.
right, which should control the phenotype, but sometimes that dominant allele just doesn't do its job.
Hmm.
Like there are times that you could have a dominant allele in terms of your actual genetics. You
Hmm.
have the DNA sequence that codes for that particular dominant allele, and yet you
You just
don't
don't
show
have
the
it.
dominant trait. Like if we were talking about the white and red flowers, you could have a red allele, and yet the color of your flower is white.
Mhm.
Like this to me is like, are you serious?
Right, like we've done all
Is
this
that
work
an
to figure out the
option? Like,
genotype, and it's just like the phenotype's like actually no, like I don't want to do
actually,
that.
no. It's like the red called in sick that day. The red allele's like, you know what?
Nah, I'm
Just
done.
white's fine.
I'm
White's
done.
fine. Like
Yeah,
I'm just gonna hang out back here.
yeah. Mhm.
This is really common, or it's not common in the way that it's tested, but again, these are all things that demonstrate the complexity of genetics.
Mhm.
And one example of this that you may have heard of, right,
Mhm.
of incomplete penetrance is actually the BRCA1 mutations, right? We know that there are certain mutations that are heavily associated with particular types of cancer,
Mhm.
but there are people that have dominant mutations that often create specific types of cancer, and yet they never get cancer,
Mhm. Mhm.
right? I could have a dominant allele that would lead me down a particular path, in this case to breast cancer, but I may not get it. And that's
Mhm.
because that dominant allele has incomplete penetrance. It may not show up. It may
Mhm.
not control your phenotype. So again, especially when we talk about genetic diseases or disorders or things that can significantly impact your life, I think penetrance is hard. Even if you know you have a copy of a allele that should drastically change your life, even then it becomes a watch and wait because it
Right.
could be there and never control your phenotype.
Mhm.
So penetrance, there's a lot that we could talk about there.
Right. Right.
Pretty topical though, by the way. Variable expressivity, incomplete penetrance, plus the last two exceptions that we're going to talk about. These are all like maybe they show up as an answer option A through D, but like even if they show up, the chances that they're going to be the right answer is probably lower, right? Like these are
There'd probably be a
lower
really
yield,
good reason
but
to for it.
cool.
Yeah,
Yeah, yeah. Important to understand, but
right.
these things, because it's basically all bets are off, just see what happens.
Just see
They
what
don't
happens.
really test them that
And
often.
if anyone's like really curious, like, wait a minute, I have the genotype. Why are you not? Like we like looked into this and it is really complicated, but some big like pointers, big directions that we're actually going to dive into in just a second are like things like epigenetics, where
Mhm.
you because again, it's more than just what's the code in your
Yeah.
DNA, but how is your DNA shaped? What modifications are there so that is it expressed or not expressed? And then there are environmental factors that change that. So even though you have that sequence, maybe that area of the DNA is super wound tight that even if you have that sequence, it's just not being expressed because of how tightly it's wound together. So that's one factor. There's other factors like, other transcriptional controls that, again, influenced by environment, things like silencing RNA, miRNA, like those things now have a factor as well. So it's all of these things kind of playing together. Why, for anyone listening, you're like, why am I have the genotype yet? It's still not expressing in that same way. Those are factors that can influence it. Again, is there something to memorize there? No, but to understand why that it may come up in the actual part of a passage, but testability wise, not really. This next one though is kind of related to this idea of epigenetics, where it's kind of like related to how like tightly a DNA is wound together, and that's the idea of imprinting, which I think is a really cool idea. I think it's shown up. There's, I think, one question in one of the exams about
I think, yeah.
imprinting.
Mhm.
And I think it's just really cool, this idea. And I know you also really like this idea too,
Mhm.
where we can have an allele, we can receive an allele from my mother or my father, but due to this idea of imprinting, because maybe I received this certain allele from my mother, just because I received it from my mother, in my mother, that allele was turned off. So because it was in my mother and I received it from my mother, that was a little turned off. If I had the exact same allele from my father, just because it was from my father, it's not turned off. So
Mhm.
imprinting is the idea that based on who you receive it from, that allele might be silenced. It's being imprinted, which I think is cool. So an example could be maybe this is some heritable disease where, oh, I only get it from fathers and not mothers, because in mothers, that allele was turned off. It was imprinted, whereas in fathers, it wasn't turned off. So when I received it, right? Oh, when I got it from my father, it was on, so I got it. Whereas even though I got the exact same allele from my mom, because of usually epigenetic modifications turning it off, it's not actually active in me. Which on a phenotype thing, like on a Punnett score, is a little bit hard to kind of show, but there would have to be something in the passage to kind of show, like, hey, oh, you only got it, you only inherited it from fathers, for example, kind of deal.
Yeah.
Yeah.
The reason why I think imprinting is so cool, in your example, you had a maternally imprinted gene.
Hmm.
Imprint means to silence. So if you
Yeah.
have a maternally imprinted gene, it means mom's copy is silenced. What I think is really cool is if you got an allele from mom, it's going to be silenced if it's maternally imprinted. Okay, so you will not express that. But when your germ cells undergo meiosis, they actually reset imprinting
Mm.
patterns to the point where mom gave you a chromosome and it was silenced because it came from mom, but you have that, you still have the DNA for it. You're just not
Sure.
using it. Before you make gametes, your body resets all of that. And now, if mom's original allele now ends up in one of your gametes, it will be active. It will
Mm.
no longer be maternally imprinted because you are a male and you
Yeah.
can now pass it on to your offspring. So
Mm.
it's absolutely fascinating. The
Mm.
body keeps silencing one or the other parent. Okay. And there are
Mm.
paternally imprinted genes as well. Um, it's just weird. It's really cool, but
It's
it,
so cool.
it will always be silenced either on mom's side or dad's side, even if you got it from mom. It's cool.
It's
Um,
cool. The idea of imprinting is really cool. And
yeah.
and that's again, why are we talking about it? It's because it's this idea that even though you have the genotype, all of these are, you have the genotype, but the phenotype is impacted, right? The phenotype is not what you would expect based on Mendel's laws. It's a little bit different. This is a great example that even though you've got the genotype, you might not express because maybe it was imprinted. Maybe it's silenced now.
Yeah.
Another great kind of exception to this, and I love this one. I think this one's really cool, is the idea of mosaicism. And you kind of referred to it earlier. It's this
Mhm.
idea that we want to come back to that for males, we are hemizygous in the sex chromosome. We have one X, one Y. But that's kind of like typical for the sex chromosome. You only want in the human body one active copy of a sex chromosome. One X, and for males, one Y. However, in females, they have two X's. Right. And so, because ideally we only want one active one, in females, one of them is turned off. And this is the idea of the formation of a bar body or X inactivation, where one of the X's, completely randomly, by the way, are turned off, which I think is such a cool idea, where in the female body, every single cell has maybe I shouldn't be so extreme. Most cells in the human body have the exact same chromosome, exact same chromosomes, exact same genotypes. Yet, in each one of a female cells, it could be a different X chromosome that's active. It could be, so in, for me, it's all the same X chromosome that's active, but in you, Molly, right? Maybe this forehead cell over here, like your dad's X is active, but maybe in your nose cell over here, it's now the mom's active, and oh, in your heart cell, it's the dad one, and the one right next to it's the mom cell. And so this is the idea of mosaicism, where females, biological females, have this mosaic of X activation. It could be the male's X, it could be the female, the father's X, or the mother's X. And so it's this different pattern. Because it's random and because there's so many cells, generally it's like 50-50 because of just variability. But there are really interesting cases where just because of variance, it's actually more of one X than the other. So you can have a situation where you, again, because in females they have two copies of the X, maybe they have one X that has a dominant like wild type normal allele and one that's recessive and that's the disease allele. And even though they have a dominant form, just because of variance, that more cells of their body like silenced the dominant healthy allele, that now, even though it was recessive, you will see some of those recessive disease traits, even though they have that copy, because more of the dominant healthy one was silenced, which I think is a really cool idea.
It's really cool. And I feel like it comes back to the size difference. Again, we
Hmm.
said Y chromosome is pretty tiny. Like
Yeah.
there's more than one gene, but really that SRY gene is what the Y chromosome is for the
Hmm.
most part, right? The X though has a lot on
So
it.
many.
Like
Yeah.
there's a lot. We talked about X-linked color blindness. Like
Hmm.
why do we have color blindness genes on our X, right? Our X is pretty big. There's a lot of different genes on that chromosome. This is also, and this is one of my favorite applications, this is also how you get calico cats,
Hmm.
right? A calico cat, it's got white on it, but it's also got patterns of especially like brown or like orange and
Hmm.
black. And every calico cat has this different pattern of spots, essentially, or splotches of color on their bodies. Fun fact, all calico cats are female for the most part. There's weird genetic things that can go on that are incredibly rare, but for the most part, all calico cats are female. And it's because the calico patterning is a visible symptom or like illustration of X activation, right? If a particular cat has a brown or orange fur allele and a black fur allele that's linked to the X chromosome, you're going to see splotches of both depending on where or which X was inactivated in that region. This, I think, is even the coolest part.
Hmm.
We've had a couple times throughout history where we've been able to clone an animal, like Dolly the
Hmm.
sheep, right? And
Hmm.
it's the exact same DNA. It's the exact same everything. If you cloned a calico cat, you would have a cat that does not have the same spotting pattern.
Hmm.
That pattern of spots has it's not genetically determined in the way that you would normally think. There's no
Hmm.
gene that says spot on your forehead, brown spot on the back of your head beneath your ear. That is all so random due
Hmm.
to random patterns of X inactivation that if you cloned a calico cat, it would have a different pattern of random X inactivation. It may still be calico, but the spot patterning is totally different. I think that is so cool.
It
Could you
is
imagine having a clone that looks nothing like you? Like,
nothing like it, right?
huh?
It's wild, but it's because of that random variance, which is such a
So
cool
cool.
thing. And again, whenever I like teach it, like I think mosaicism actually comes up like more than once in questions I've noticed. I think there's like, not again, I'm not the highest sealed thing in the world, but it does come up in some questions. Again, I just think as an idea, it's really, really cool.
Yeah.
And I think just genetics in general is such an interesting field in terms of the MCAT in general, and also one that we need to understand, especially going into a field of medicine, and especially like in the future of medicine. A lot of medicine is leaning towards a lot of like looking at like tailoring someone's medical treatment to their genetics. This is already something that's happening in today's like I remember like working in a pediatrics clinic. They would take like the gene like the genetics of like the genome of a patient like of children in order to determine which medications would be best suited for them because they might have some enzymes that break certain medications faster than others. So that's how they determine that. So this is a growing field in the future of medicine that I think is, more than just the MCAT, something that's really important to understand. And I think on that kind of field, right? I think we both talked about this, Molly, that we really want to hit on like genetics, a lot on genetics for the next few episodes.
Yeah.
Uh,
Yeah. There's so many really cool things about it. And you're talking about like how this is going to impact medicine. CRISPR-Cas9. If you've never
yeah.
heard that, go Google it.
Yeah,
There's some
it's
really
huge.
cool books. It's actually one of the books on my bookshelf,
Hmm.
Codebreaker. Pretty awesome. The idea of CRISPR-Cas9 is insane. It's like
Hmm.
basically well, I won't get too into it. It's a tool that will allow you to do incredibly precise
Hmm.
genome editing. Like the implications in terms of medicine, but also moral and societal
Ethical
implications,
things,
ethical
yeah.
things, it's fascinating. But like we are in a time of such incredible genetic discoveries that like you need to be comfortable with this if you're getting into medicine. Anyways, I couldn't help myself because it's so cool. But yes, I definitely want to talk more about genetics. I feel like we've talked about something that's really critical and underlies a lot, but in terms of how genotypes create phenotypes, there's so much more to be said.
Right.
Specifically, we need to talk about how we make functional products from our DNA, right? Like DNA is not controls your phenotype directly. DNA has to code for RNA, which then codes for protein. Proteins do most of what the
Ev-
DNA
Every,
encodes, right?
yeah.
So we need to talk about central dogma. And even more than that, we need to talk about how gene expression, right? So transcription, translation, things like that are regulated. Because
Controlled.
like you mentioned, every cell in your body, for the most part, has the exact same genetic makeup. And yet you have photoreceptors in your eye that detect light. You have keratinocytes in your hair, skin, and nails that are mostly for protection. You've got hepatocytes in your liver that are carrying out so many unique functions. Like they all have the same genetic code. What matters is which portion of the code you use, to
Hmm.
what extent you use it, and how you time the use of that gene during development. So, gene
Hmm.
expression regulation is critical too. I'm very excited to talk about all this good stuff. So, that will be,
Next episode.
yes, that will be what we talk about next episode.
Right, right. And I think it's super, super cool because, again, like you said, we've kind of hit big picture on how genotype leads to phenotype, but now we're going to zoom in on the mechanism. How does this blueprint that's in every one of cells actually get used? Because really, that's what DNA is like the blueprint. Our bodies are protein machines. So we're going to be talking about how it goes from blueprint to protein next episode. So get excited. There's going to be a lot of really, really high yield stuff next episode. Not that like today wasn't really high yield, but next episode, very, very testable information next episode. So stay tuned for then. But in the meantime, happy studying, everyone. And I hope wherever we're finding you, you're taking care of yourself and enjoying the time.
Bye, guys.
Take it easy. Welcome to the Jack Westin MCAT podcast with your hosts, Michael Maleka
And Molly Hudash.